Good afternoon. Good afternoon, sir. Can you hear me? Good afternoon. Good afternoon, Dr. Palanikumar. Can you hear me? Hello, sir. Good afternoon, sir. Can you hear me?
Yes, I can.
Yeah. Yes, sir. Yes, sir. We'll start now. Good afternoon, everyone. Welcome to the last and the final session of our international webinar on biotechnological perspectives. Today with us we have Dr. Palanikumar Manoharan, who's from PPT Global Central Lab, Clinical Research Organization, Highland Heights, Kentucky, USA. Today he is going to talk about modern biotechnology spearheads, the global fight against COVID-19. Before we get into the session, I would like to first introduce our guest of honor today. Sir did his master's in microbiology at VHNSN College, and then further he went on to do his PhD in medical microbiology from University of Madras. His research field is streptococcal infections, and further he did his postdoctoral fellow in West Virginia University, where he did his work in inflammatory bowel diseases. He was also the research associate at University of Cincinnati, where he did his work in cardiovascular and skeletal muscle disease under the guidance of Dr. Jerry B. Lindgren. Currently, he's working as the senior research scientist at Molecular PPD Global Central Lab, Kentucky, USA. To talk about his publication, he has quite a number of publications with high impact factors. Some of them I can tell you are Circulation Research, JBC, American Journal of Physiology, and iScience. Sir has also presented many papers in various international conferences. And to talk about his professional affiliations, Sir is the associate editor in journals like Nutrition and Metabolism. He is also the member of Association of Molecular Pathology and member of American Society of Microbiology. With this very short introduction, I would like to welcome you, sir, to this session. Thank you very much, first of all, for accepting our invitation and sharing your valuable time with us. Welcome once again. I now hand over the session to you. Please, sir.
All right. Thank you for introducing me to the crowd. I thank Dr. Sham for inviting me and giving me an opportunity to present in front of my hometown students and faculty. So it's an honor to be a part of this program. And Dr. Sham, I have never had given a seminar before 6 a.m. And thanks to you, and you're so special. And I take this opportunity to thank you for that. So, can I start, Dr. Sham, with the slide presentation?
Yes, sir. Yes, sir.
Okay.
Yes, sir.
Okay. Let me know if there is any trouble in seeing the slides. I can restart it. I hope you can all see this now. All right. I chose a topic slightly different than what I normally do. My topics are usually based on clinical and medical microbiology, totally based on the pathogens and species barrier breaking, epidemiology, things like that. Today, I took a little different tone as requested by Dr. Sham. He wants to have a small input of what is this current status of the vaccines and how biotechnology could play a big role in this COVID-19. So if at any point you have any questions, you can stop me, or we'll have a last few minutes to discuss over this. So since due to the time constraint, I have given a very few examples here down the line. You can see in some of these slides. This is just a representative that I'm going to give, figures I'm going to give. It may not be the real trend of what's going on. So, but however, you will have the core information, and we'll move along. So this was clicked a few hours ago before I had prepared. This was the last slide I made. So you can see this John Hopkins created a website. They update this every second around the world, the number of cases and number of deaths. So you could see this red all over the globe. So this kind of indicates that yes, it is a pandemic infection. Close to 6 million people are affected, and 365K people died so far. So, which is an alarming thing, and it is not going down. And if you can see, countries like Brazil and US, who reported this late, are picking up this infection pretty fast. Brazil are already close to 500K, and Russia is closing to 400K. This looks like it's this respiratory infection is never going away without a fight. This is a figure from India. This is again few hours ago. We have 175K infection and close to 5,000 deaths. This is really a very grim and bad situation here that we are facing across the globe. Not able to do our routine work, not a routine life that we had before, not the normal way we used to go out with friends, family, and in the workplace. So we have restrictions all over the world just because of this one virus. Are we new to pandemics? No, we are definitely not. This is a visual capitalist figure, so I just took this figure so this could be more precise, and I can bring everything in one slide. So are we new to pandemic? No, we are not. We have been used to this major pandemics over again for the past few centuries. So this is not new. But what we are seeing here is there is a pattern of evolving of this particular coronavirus. So earlier, the first outbreak, it started with a SARS in 2003, which claimed close to 1,000 lives, and then we had an outbreak of MERS in Middle Eastern countries. Again, the death toll did not take a big this thing at that point. They kind of controlled it, and now we have a mutated virus and very virus that could replicate really fast and which has a robust uh, uh, stability in environmental conditions. That makes us all scary. Like, why would they stay in the environment for such a long time? Usually, a cold or a flu viruses that go away pretty fast. So we do have seasonal flus, and they do tend to uh, behave according to the climate. During the warm weathers, they kind of dry away. Then enzymes that are required for infecting the humans, they're not active anymore, and they die. But very unfortunately, this respiratory virus, this mutated new coronavirus, has the property which could stay in the surface for a longer time, and the replication rate is pretty fast. So we'll talk more about this virus shortly, and then we'll move on to this biotech part. So these are the different coronaviruses that has been known to affect the humans. And if you can see these four, like 229E, OC43, HKU1, NL363, these are the common coronaviruses we have been testing so far in a respiratory laboratory. So you could see these viruses coming, getting humans getting infected because of these viruses quite often. And then we have SARS-CoV and MERS-CoV, which had an outbreak in the past decade. And then apart from human, there are a few other species who could be potentially intermittent or host for transmitting the disease. The civet, cow, camel, and bat. These are the most common thing, and I think they missed the pig, swine here. Swine is again intermittent host that could transmit this coronavirus into human population. So again, this species breaking barrier, species barrier had been broken several times in the past, and this time was really strong, and the virus is highly potent. SARS-CoV infection and COVID-19 pathogenesis in organs. So let me rush through this quick, so that you get an idea of the nature of this pathology. So there is still a lot of debate going around the world whether this virus was generated from the lab or it was contaminated from the lab into the public, things like that. But so far, all the scientific evidences and the gene sequencing data and the cluster they have traced so far point to that it came from the bat, and the most possible intermittent host could be the pangolins, which is a Malayan pangolins, which was found in Guangdong, China. So by natural selection is an animal in an animal host before zoonotic transfer, and then again it took a natural selection in humans following the zoonotic transfer. So that's the pattern we have been seeing so far, and I'll be a little bit giving a hint about that down the slides. Right. So there are all over the world, they are fighting to study about this virus and know what is the most important thing about this virus. SARS-CoV-2 is an RNA virus. It's a single-stranded envelope virus. The most important protein that has been linked to human infection is the S protein. So this is the highlighted region here. You could see the entire graphical illustration of this S protein, and it has two subunits, S2 and S1. And the S2 subunit, which is all covered with the carbohydrate surface, I think that they believe that could be one skipping these virus from the immune system. And the S1 subunit has three domains that is known to attach to the human ACE2 receptor. And most interestingly, the spike protein opens up when they are ready to bind to the human cell, and this opened spike protein binds to the ACE2 receptor. So this is a little bit background about what happens when they bind to the ACE2 receptor. So when I'm sure you might have all heard about this pathogenicity of virus and how they enter over. You must have heard this word ACE2 receptor quite some time. So angiotensin 1 is a peptide that is being released from kidney during any stress or infection or during the metabolism. So this angiotensin 1 is converted into angiotensin 2 by an enzyme called angiotensin converting enzyme. So these angiotensin 1 and angiotensin 2 are vasoconstrictor, which means that they have the potency to increase the blood pressure. So angiotensin 2 has its own receptor. When there is a massive amount of angiotensin produced, you end up with an acute myocardial remodeling, acute lung injury, and vasoconstricting events, which increases the blood pressure. So what naturally our body does is it has another enzyme called angiotensin 2, ACE2, angiotensin converting enzyme 2, which converts angiotensin 2 into several different subunits, angiotensin 1, 2, angiotensin 7. And thus they prevent the vasoconstriction or injury, acute lung injury. So people who doesn't have who has an increased ACE production, who has an increased trouble because of this angiotensin and increased blood pressure, you must have seen them always having these drugs like losartan and lisinopril. So there are recent studies that are indicating that people who are having these two drugs eventually ended up producing more ACE2, which is again making them vulnerable to SARS-CoV-2 infection. So there is an increase in ACE2, and eventually there will be an chance of getting this infection is higher. This particular information was got from New England Journal of Medicine, which was published in April 23, and they have quite some details about this two drugs. But here I just want to show you the mechanism how this virus could enter the body. Please bear with me. I'm having trouble moving to the next slide. Can you all hear me though?
Yes. Yes.
Yeah.
Yes,
Please bear with me. I'm having trouble moving to the next slide. Hold on.
Okay, no problem. You try to.
All right, there you go. So the occurrence and outcome of severe acute respiratory syndrome, coronavirus 2 viral sepsis. So this is something we all need to be aware of. There are three different kind of people who are being infected with SARS-CoV, and they present with the three different clinical conditions. One is severe infection we all know that what happens to people who are being having an acute SARS-CoV-2 infection, and people with mild infection. The majority of people who are infected in India have recovered, and they all had a mild infection. And the most watchable and the people who need to be need to be diagnosed, and we might miss them, is the asymptomatic people. And this figure here shows what the difference between what will happen when you have a mild infection compared to the severe infection. But during a severe infection, there is a massive infiltration of inflammatory cells that's coming into your lung, and which damages the lung severely. And with the mild infection, you may not have that much of immune cells that comes in, and then eventually your natural immune system kicks in and prevents the further damage of lung cells. So that's how people recover. Good. What happens to people with asymptomatic infection is they could carry the virus very well. They may not present with the fever or cough or any respiratory distress, but they will be spreading the virus to people who are elderly, who have metabolic disease, who have underlying health conditions, and make them vulnerable to this infection. So that those are the people who need to be watched. So I think government in all the countries are now trying to set up a testing platform for people who are asymptomatic, so they could test before they go back to a job, or could test before they could get out of this lockdown. So by this, what we can do is by screening everyone, the possible people who are being exposed to SARS-CoV, we can keep a healthy environment around. And these are some of the slides which explains the severity of infection. These were obtained from patients, postmortem of patients who died because of SARS-CoV. So if you can see on the first slide, it shows the mononuclear and macrophage infiltrating the airspace and diffuse thickening of alveolar wall. And here again, you see an formation of Highland membrane, which is severely impaired as a blood and tissue oxygenation is impaired. And RNA in situ hybridization was done on these samples and postmortem lung, and you could see this severe infiltration of virus that is lining the alveolar. So viral particles were observed in bronchial and alveolar cells, and patient showed hypoxia, spleen atrophy, lymph node necrosis, hemorrhage in the kidney, enlarged liver, degeneration of neurons in the brain. What not? I mean, there is no limitations for this virus. It was seen everywhere, and every clinical sample they collected, the RNA of this virus was seen. Urine, feces, blood, saliva, bronchoalveolar lavage, whatever samples you take from the patients, you still were able to see this SARS-CoV. And that's something strange about this virus. Usually a respiratory pathogen will never cross the barrier of intestinal barrier to go into the GI system. So when you can isolate this virus from stool sample, which means that this virus is really resistant to all acidic conditions, it could pass that. And what makes this virus uncontrollable? Like, why people succumb to this infection and cannot recover so fastly? Like I mentioned in the previous slide, you have a massive pathological conditions which people try to tend to repair with their immune system and get back to normal. And then you have the hypoxia. Hypoxia by itself can produce inflammation. On top of hypoxia, there is something was observed in this SARS-CoV infection that's called cytokine storm. Cytokine storm is nothing but a collective cytokines that is being released massively by the immune cells that come into the lung to prevent the infection, but eventually they fail, and then they break down, and all these cytokines and chemokines are released from these immune cells. And if I remember very well, sometime in April, first week of April, WHO kind of mentioned in their website that they had a meeting about this, And there was a research group who worked on IL-6 to tackle IL-6 and see if that could rescue the patients from this infection. No, they completely failed because though IL-6 was the massively produced cytokine, there were other chemokines and cytokines released from this virus, which made it very difficult for them to treat just with the IL-6. So they kind of stopped that program completely to move on to a different target. And one other strange, abnormal thing that you may not see quite often in news is like 71% of the non-survivors of COVID-19 showed an abnormal coagulation. So what happened is the epithelial endothelial barrier was severely interrupted and attacked lung capillary. It attacked the lung capillary in the endothelial cells. So now you could understand why people had a respiratory distress on top of this infection. They couldn't recover very well because the capillaries that was broken in the alveolar and the lung epithelium kind of had a blood coagulation, which made them put an enormous stress in their lung. The current treatment options are these four right now around the world. Remdesivir has been proven to be successful with the Ebola virus. So they believe that this would be the next possible drug that is coming into the market for a quick treatment around the world. And lopinavir and ritonavir, which are licensed HIV drugs, have been tried by several countries. Interferon beta 1A, which is used in multiple sclerosis, has been tried. Chloroquine and hydroquine, which is used to treat malaria, has been used as a prophylaxis and the treatment. You must have heard about this possible troubles and side effects of chloroquine and hydrochloroquine. Regardless of that, there are some positive news about these drugs that has been used. So now let's focus on the biotechnology part. Why we had this modern biotechnology taking this whole thing in hand and running faster than any other medium that could solve this trouble. So recently in London, there was a study made from they tried 7,500 strains were sequenced, and they found 200 mutations, recurrence of repetitive pattern of mutation in these viruses. So we are kind of this virus is totally a new virus. Like, nobody knows how it behaves, what needs to be done. And the biggest challenge we had is the time, because we didn't know anything about this virus. We didn't have any available testing capacity for this virus or any treatment that we know on top of our head that will work on this virus. So everything was new to us, and the time was the biggest factor. People were suffering, people were dying, we were not able to help, and because we didn't know how the transmission was and how potent they could stay in the surface. So, I mean, thanks to the biotechnologists and people like you who have taken this in the hand, and they're taking a massive lead at this point, since they know the sequence of this virus and property of this spike proteins. So, let me talk a little bit about what are the different kind of vaccines they have tried, and all these biotech companies who are leading this fight against COVID. So there are 4 different categories of vaccines: viral vaccine, viral vector vaccine, nucleic acid, and protein based. So these are the number of companies, number of people who are trying to work on this virus. Close to 90 different companies and university and private sectors had joined this race. However, there are very few people who are working on a virus by itself compared to protein based and nucleic acid based. So what is the viral vaccine? There are 2 different viral vaccines, which is a weekend vaccine or an inactivated vaccine. Weekend vaccines are usually called as live attenuated vaccines, so which could have a potential to elect the proper immune response to that virus, specific virus that we are targeting. The case is SARS-CoV-2 and inactivated vaccine. So this is a diagram that shows how these vaccines are used. If you can see, the weekend vaccine enter the cell, they do go through the normal replications. By genetic engineering, they would have reduced the virulence or modified the virus not to produce a real blown out infection. And the properties modified could elect a proper immune response through antigen presenting cells, and then we have protection for lifelong. But the trouble here is immune response is not good, and if you have antibodies that could affect the pre-immune antibodies that are present in our body, that could affect these virus directly, and we may not have a good immune response that stays for a longer time. And inactivated vaccines or heat killed vaccines. So what happens is, even in my lab, my own lab, we have inactivated SARS-CoV-2. What we do is we get what they do is they put this virus in 65 degrees Celsius for 30 minutes. They lose all these property. So immediately it comes from BSL-3 to BSL-1 safety level. We can use that as a spiking controls for RNA extraction or downstream processing. So this inactivated vaccine had been shown to shown a protective effect in monkeys. So the Sinopharma, which is also known as the Sinovac, they quickly moved from phase 1 to phase 2 because of the positive response they got. So we might be having a news coming out of this company very soon with their data on the phase 2. And on the weekend vaccine, here again, the Coagulation, which is known company who can do the genetic engineering of all these live viruses. So they joined hand with the Serum Institute of India. And I mean, there was a article about this from Serum Institute saying that they could produce as high as 100 million vials if this has been shown to be effective. But right now they are at the preclinical stage. I am not updated on their phase right now. And the second category is the viral vector vaccines. Again, here there are two different categories. There are replicating viral vaccines and non-replicating. Here is where the biotechnology and genetic engineering plays a big role. We need to understand the system very well to quickly, the first case of corona, the WHO was reported was 31st of December, where China informed WHO that they had an uncontrolled infection going on. And we had a first death due to coronavirus sometime in February. And now we are here in May. There are 90 companies who are already working on a vaccine with various different vectors, various different forms of these vaccines. So this is, I think, this modern biotechnology is very powerful tool that we could tackle any infection in the upcoming years. But this is a good lesson to learn for us how to tackle the pandemics and outbreaks like this. So the replicating virus again takes intracellular route, and non-replicating virus could directly be ingested by the antigen presenting cells, and they could elect an immune response. So here both these viral vectors will be encoding spike gene. Either it could be a spike gene for S protein or M protein or N protein. Any of these SARS-CoV virus protein that is being known can be used as put into this viral vectors. And this has been pretty successful. I mean, recently the Ebola virus got an FDA approval for using a replicating viral vaccine. So they used something like a measles vector, which will have the target genes engineered into them, and those will elect a proper immune response which will protect us from the viral infections. And again, people who work on this, there are a lot of research going on with the non-replicating vectors, because those viruses are very easy to work, because they do have a property of common cold viruses like adenoviruses, or the most common non-replicating virus used for this kind of vaccine production. So adenovirus is a kind of common cold, produces a common cold. It itself has a property to do that. And Johnson and Johnson had recently joined the race with the adenovirus 26, and CanSino Bio. To my knowledge, the CanSino is the leading company that went to the phase 2 very fast. So while all others were in the preclinical and the phase 1, CanSino Bio, I think it's the Chinese-based company, they moved to the phase 2 really fast, and they have pretty supportive data, which might go into phase 3 very soon. And protein-based vaccine. These are again, you need you all know how a protein-based vaccine works, because they take the protein, the protein has an adjuvant, and that is injected into the patients, and that will elect a response. Again, this is under a phase 1 trial going on. Most of these companies around the world, most of the educational institutes around the world are using a protein-based vaccines, because it's pretty easy to work on the in vitro and the in vivo model in the lab. So they prefer this route, and the immune response is pretty strong, and it is stable. So the research labs and biotech labs who have experience working with viral vectors will prefer to work on the protein-based vaccine. And this is an example here, a Novavax. They are leading this protein-based vaccine research, and they have a full recombinant SARS-CoV glycoprotein, which is in a nanoparticle-based vaccine. Vaccine for influenza infections. So this might again turn out to be a very positive vaccine, and everything is in a pipeline and everything is in testing mode. We don't know the exact timeline when they will come out. And the most important vaccine of modern biotechnology is the nucleic acid vaccine. So though this has been under practice for a while, and now we have evolved so well that even a small piece of DNA or an mRNA is enough to elect a strong immune response. So this picture again describes how these vaccines are given to the patients. And RNA vaccines are encapsulated in the lipid coat, and whereas DNA vaccine can be injected directly as a naked DNA into a host body through electroporation. I'll be talking about this DNA and RNA vaccine in the upcoming slides. And here again, I picked one example who has an established model to deliver the DNA vaccine. Innovia again is leading the DNA vaccine study. There are several other institutions who are working on DNA vaccines, but the electroporation delivery technology that is patented by Innovia is pretty handy at this time. So what they do is the naked DNA has a small injection here in this machine, which will go into the subdermal region and release your vaccine. They are 1,000 times more effective than your other routes of DNA vaccine dosages. So we have seen respiratory aerosol vaccine, drops, IM, and several other vaccines. This is something new that technology, we had to capture, than electroporation based delivery. So this might again be a very successful model. The San Diego based company who are leading the research on DNA vaccine. And this is, I saved this one for the last, because you might have heard of the news about this company, Moderna. Moderna used mRNA vaccine, and it's called mRNA 1273. They did a lipid nanoparticle encapsulated mRNA. This mRNA will code for this viral antigens, and which will eventually elect a strong immune response. It will mimic as a viral infection. So last week, I think the third week of May, there was an breaking news from Moderna. Their first phase 1 results were out. They produced neutralizing antibodies against SARS-CoV with this particular vaccine. So, which means that the company news said that their antibody titer was so high as if a patient was really infected, which is a good news. And they were able to produce a neutralizing antibody, which is again a real good news that will be a very safe vaccine and very effective vaccine in the upcoming years. I mean, I'm assuming that their first vaccine would be coming out in early summer 2021, but the timeline depends on the phase 3 and the final testing going on. The phase 2 and the phase 3 going on. So they cleared the phase 1 and they moved to the phase 2, where they are recruiting more patients to this, volunteers and donors to that, so which will have a result very soon. So we all know this, like, 90 different companies are working, and several academic institutions are working on this. Why there is a delay? Why can't we have a vaccine right away? Because that's the world needs this at the most. Why are we not having this? So usually when you have a clinical trial, it has to go through several phases to get it approved. Though there are emergency approval going around the world because of this pandemic situation, but still they have to go through all these phases to clear the drug to be given to the market and for the usage of people. So it goes from preclinical to phase 1 to phase 3, and you can see here the number of participants who are recruited in each phase. And most of these companies, biotech companies, are in phase 2, which is a good sign actually, because if they had an adverse effect, they would have seen that in the preclinical and the phase 1. And once they have an adverse effect and not able to pass the phase 1, then that's going to be a big setback, and it's going to take several years for us to clear. Once they move to phase 2, which means that their vaccine is safe, now they're going to look into the titer and efficacy and the stability of these vaccines with a larger number of participants. So we are all having the fingers crossed, waiting for these vaccines to come out. And these kind of webinars are to create a thought-provoking ideas. Now you know what are the different kind of vaccine, what are the different targets we can look into. So anybody who comes up with an idea of tackling this SARS-CoV will be appreciated at this time. I think both state and central government are ready to spend any amount of funding for this kind of research and clinical trials. And I would like to complete the talk with the good news that this paper came 10 days ago. There is an unanswered question. A person who had been infected with SARS-CoV, when they're exposed to SARS-CoV-2 again, will they be infected again? Will they have will their immune system memory will be prolonged and will be stable enough to stop the second or reinfection? So the good news here is you can see, look into this paper that was released, published in Science 10 days ago. They found out from a rhesus monkey study that when they infected the 9 different monkeys the first time, it did develop all this SARS-CoV related pathology. And once the viral clearance had happened, these animals were again infected with SARS-CoV. But when they were infected the second time, the infection rate was extremely low, because the animals did have a good immune booster developed, and they were able to prevent the second infection. So this is a good news, and all these biotech companies that are taking up this, speeding up the process of vaccine production and drug production. Again, that is a good news. So we are all having a positive hope that we could all fight this virus together and have a safe future for all of us. So in addition to fighting with virus, we also have to fight with all the misinformation and gossips going around the world. So I would strongly recommend all the students to go into only legitimate websites to get this information. Either use your WHO website, CDC, ICMR, and if you want to know about the clinical trials that are going around, use this clinical trials gov site. And I thank all the frontline warriors who are all the real heroes who are saving so many lives at this point. Their selfless act is much appreciated, and each and every individual we have to thank for helping us being safe. And I thank my mentor for shaping my career, and I thank my Professor Dr. Lingral, who was my postdoc mentor in the University of Cincinnati. Thank you. I am ready to take on questions. Hello? Can you hear me?
Yes, doctor.
Okay.
Thank you.
Did I make it in time, Dr. Shyam?
Yes, within the stipulated time you have finished your talk. And the program is linked in YouTube Live. So hence the participants are not able to ask questions directly. So they have posted the question in the chat box. So that has been linked in this meet also in the chat. So please, if possible, we can make some answer to these questions in the chat box.
Okay, sure, I can do that. You want me to answer them now or later?
Yes, if possible, some of the questions.
Oh, sure. What is the perfect database for retrieving the gene sequences? I can send them the link for that. Proteinase, can this proteinase 2 and 1 can be deteriorated by the chemical ligand? So I can answer this question. Can this protein ACE2 and 1 can be deteriorated by any chemical ligand or by immunoglobulin? That is a pretty risky thing to do. Like I mentioned, ACE2 helps converting angiotensin 2 to angiotensin 1 to 9. Angiotensin 2 is a vasoconstrictor. If we are going to target ACE2, your angiotensin levels are going to go up. People who are having cardiac problems and predisposing conditions in terms of pressure will have trouble. So that's why it's like a double-edged sword. You cannot have them more. You cannot inhibit them beyond a level. So that's one other reason a lot of people having heart conditions died because of this. So I think there are some research going on, but to find the dosage level where they could have a protective level of angiotensin 2, at the same time prevent the viral entry. So we might have some publications soon coming out on that. And why does this virus takes 14 to 21 days for the expression in body? I would say, like, to that matter, most of these viruses do happen, except the seasonal flu. Most of these virulent viruses do take 14 days of time. I doubt if it is 21 days. So far, they have found it is between 10 to 14 days you can have the symptoms. So what happens is the viral load that comes into your exposure might be very low. So once they capitalize the mucosa, they start replicating. They kind of adapt into the situation, into your intracellular situations. Once they find their niche, once they find their favorable environment, they started replicating faster. So to that matter, any virus that infects this kind of potent virus is pretty dormant at the initial stage, and then they go faster. And they need an acidic condition to have this exocytosis going on. To get get out from the cell, they need an acidic condition. So they kind of wait for all these favorable conditions to kick in for this rapid replication. Know that the doctors are trying to utilize plasma therapy treating COVID-19. Oh, that's a very nice question, actually. In fact, I want to make a comment on that. So the question is, the doctors are trying to utilize the plasma treatment for treating COVID-19. Yes, this has been going. In fact, I myself asked this question to a clinician here in Cincinnati during a webinar. So his answer was, this answer is from a clinician, not from me. His answer was like, they do treat the patients, COVID-19 patients, with plasma at the end stage. So if they have no other choice, like they cannot treat them with antiviral, they lost their life 85%, there is no other hope, then they go for this plasma treatment, because you never know what adverse reactions they will create when you take a plasma from a patient. And you will have minimum time to screen those plasma and then give it to these patients. So the risk is too high, because having the blood-borne diseases and blood-transmitted diseases high, it's quite risky. But they do consider that for patients who are gravely ill. And anti-inflammatory components are being used to COVID-19. How far it is possible? Yes, you're right. Anti-inflammatory components are being used. Like I mentioned in my talk, anti-IL-6 was a total failure, actually, because that was the first project they picked up. They thought if they block IL-6, they could completely stop this number of deaths and this high pathogenicity of this virus, but they couldn't do that because, again, the cytokine storm that comes in is because of the massive infiltration of your macrophages and neutrophils. When you have this much of severe infection, it's very hard to just use an anti-inflammatory as a treatment regimen. You need a multiple drug to be given at this point. How much is possible to eradicating the COVID virus through the CRISPR technology? That's going to take some time. Yeah, that's a very good question again. CRISPR technology is good. It has been used worldwide. A lot of academic labs are trying to come up with that technology too, but so far there is no data available to prove this efficacy of this technology. So it would be very wrong for me to comment on that too early, but that's a very nice thought. Almost all are taking COVID-19 irrespective of their education level of field and specialization. This is an exceptional presentation. Okay, thank you. Sorry. When will the COVID-19 pandemic end in the world? And guess prediction. There is no prediction to end this. We don't even have a prediction how we are going to treat it and how long it is going to have a perfect and safe medicine. So the SARS-CoV is going to be in our community for a long time, quite a long time. You know how they eradicated polio, how when they started the polio vaccine, this thing, we are still fighting with that. And how we fought the smallpox virus. We kept the smallpox virus away from the community. Similarly, they will do that with the SARS-CoV-2. The trouble here is like these are viruses that came into the human community, breaking the barrier. We will never say that will in another 10 years we might have a new SARS-CoV coming in from the bat. So I should have again a different, because I had a one more presentation in a college in Coimbatore where exclusively I spoke only about the viruses, how they break the species barriers. That's a quite long topic. Probably I could send some links to Dr. Shyam and he can share it with you. It's the community by itself has to grow. We should keep away from animals. We have to respect the nature. We have to leave those mice that live in the jungle to live by themselves. We shouldn't interfere with that. So all these rules were broken in Wuhan, where this virus originated. So it's very hard to predict how we could end this. Antisense RNA therapy for COVID-19. Yes, antisense RNA could be tried. But I believe that immune response, something that will elect the immune response, and that a stable immune response would be ideal here. Antisense RNA could have some effect, but in a longer, in a very practical way, I think electing an immune response, vaccine that could elect an immune response would be ideal. Whatever the medicines that are used to cure SARS-CoV, why can't we use to cure? The reason here is the antigenicity of both these viruses are different. In fact, they say both have a same mechanism of entering the human body with ACE2 receptor. The affinity for SARS-1 was very low compared to the SARS-2. And all the sequences are pointing towards that these two are completely different virus. So the treatment may not work very good. I mean they tried the first, that was the first thing they tried. How much will the high mutational and unstable character of SARS-CoV pose challenge towards vaccine production? Yeah, that's the, I think I started my statement with that. Saying that there are they tried 7,500 sequencing, and they found more than 200 repetitive mutations in that. So it's pretty big challenging. So all these vaccine companies, that they're targeting a very conserved region. Their product is going to be a very based on a very conserved region where they don't have much of mutation noted. So I think the biotech companies are pretty aware of that. Yeah, plasma therapy, I think I answered that question. Did I miss something? Is that Ayurvedic medicine works against... unfortunately, I don't have any knowledge on Ayurvedic medicine that I could answer right away. But I know I did notice one thing in WHO website and other places, that neem spray has been officially added into WHO as an anti-SARS-CoV agent. So I have seen, I myself personally know that there are several people who are using neem extract and neem spray to keep this virus off. And I'm sure Ayurvedic medicines are good as well. I didn't do any literature or this thing on that, so it will be very wrong for me to comment on that. Siddha Ayurveda are also doing research. Yes. They are working on that. And in fact, this Kalonji seeds, I know one of my junior who's working on this Kalonji seeds, he found an active compound in that which could bind to the spike protein. which has the affinity to find to the. In fact, I myself tried that kalonji seeds a few times, and there are several other ways Ayurvedic and Indian spices that could potentially have a beneficial effect. Kalonji seeds that I think they call it as black fennel. That could be a potential option here. Did I answer everything, Shyam?
Yes, yes, Dr. Palanikumar.
Yeah.
So there is one more clarification also. One person asked raised one clarification. That is, what extent the bioinformatics tool works on the virus to find out like antibiotics and so on.
I'm not pretty sure on that.
Because nowadays there are more number of laboratories and people are working with the bioinformatics tool to find out the target.
Okay.
Like that. So, what extent it is applicable, and how far it is feasible to control this COVID-19 virus?
I would be the wrong person to answer that. I don't have much experience by using bioinformatics as a tool. But having a situation like a pandemic outbreak like this, any tool is useful here.
Okay.
That will be a general comment on that.
Okay. Thank you very much. So we hope that this session was very nice, and even as a microbiologist, I do not know about this virus. I have known only little extent about this virus, how it infects people and causes disease. So after seeing this your presentation, I also learned more how this virus enters and how it causes disease and inflammation and all. So I think this session is wonderful, and I hope that every people who listened, that means participants who listened to this session, will be a fruitful one to everyone. Thank you very much, Dr. Palanikumar. And
my pleasure. It's very strange for me to call you Dr. Shyam. I call you you're like my brother.
This is official. Thank you very much.
I believe so. It was very nice, and thanks for organizing this.
I may conclude this session. So before concluding this webinar, I would like, this is the time to give vote of thanks to every people, those who supported to organize this webinar program. So at this juncture, I would like to express my sincere thanks to the management and the principal, Kamaraj College of Engineering Technology, given permission to organize this 3-day international webinar successfully. And extend my thanks to our head of the department. She is backbone of the department, as well as to support all kind of activities in our department. So thanks to our head of the department. And I extend my thanks to the speakers, Dr. Karthik Balasubramanian, scientist, Agarkar Institute, Pune; Dr. Karthikeyan K, scientist, Gujarat Institute of Desert Ecology, Gujarat; and Dr. K. Ponmurugan, Kingsard University; and Dr. S. Shahjahan, scientist, Tata Chemicals; and Dr. S. Sudhakar, head Department of Biotechnology, Manoharan Sundarnar University, Tirunelveli; and Dr. S. Lakshmi Subramaniam, CEO of Sridhi Herbals Extracts, Coimbatore; and Dr. Palanikumar Manoharan, scientist, PPD Global Clinical Lab, USA. So I thank all the speakers who have given wonderful talk about various topics. And I extend my thanks to Dr. Geeta, who has given master of ceremony and introduction to every speakers for the past 3 days. And I thank the technical people, admin group of our Kamraj College of Engineering and Technology who supported to organize this program successfully. And I extend my thanks to Dr. I. Ganesh Murthy, Dr. S. Karthik and Ms. Amudalakshmi, my co-coordinator of this program. I thank all our department staff, teaching staff and non-teaching staff members who supported to organize this webinar successfully. The last but not least, I extend my thanks to all the participants. Without these participants, we could not organize this such a wonderful program. So I thank all the participants who participated patiently and raised questions for the clarification. Thank you very much. Thank you, one and all. Dr. Palanikumar?
Yeah. Thank you, Dr. Sham.
I'll take this total webinar session.
Sure.
Thank you very much. Thank you very much.
Yeah, thank you. And I think I missed a few questions. I'll probably answer them directly in the YouTube link if I can access to that. Thank you.
Okay. Thank you. Thank you very much. Thank you. Yes, bye.
The link will be sent in the WhatsApp group. Okay, go to the link. In the WhatsApp preparing